Well, let's shift to another topic. That was fun. That was good.
Okay.
Andrew, I know there was a few things that you were talking about earlier in terms of evidence for evolution. Everybody can get involved on this one if they'd like to. A couple of points you and Matt were going back and forth on as well, but we just didn't really have the time in that moment
not what
to
they were
elaborate.
exactly at this point, unfortunately.
Well, if you could present a line of evidence, and we'll have Matt get involved first, and then we'll kind of just go with the flow with that. So go ahead, Andrew. Andrew versus Matt, the much anticipated debate.
Yeah, well, I don't think me and Matt get to interact much nowadays, so I appreciate having me on. I guess, let's see. The last thing we were talking about was the whole synonymous mutations, ubiquitous proteins thing. So I did have a few other things I want to say about that. I guess if you want to reiterate some of the points you were making, Matt, just to kind of review some of them.
Oh, I was talking more about, I was trying to help Ken out and answering the genetic boundaries thing, which was
right.
And then you moved into synonymous. That was with Donny, actually. That you
were yeah. Okay.
If I didn't know if you wanted me to go into it or not, but
Matt, what are your thoughts on? Okay, so Andrew, correct me if I'm wrong. You've got these proteins that are shared across the biological world that fall into nested hierarchical patterns, and then the synonymous mutations within those proteins also form a hierarchical distribution. And you're arguing that those synonymous mutations aren't really doing anything specific function-wise to the organisms. They're neutral, functionally neutral, basically.
For the most part, there are some cases in certain lineages where they can be under selection to do specific things, but for the most part, they're under very weak selection pressures. So yeah, pretty much for the most part overall.
Go ahead. I gave my thoughts earlier. I'll elaborate a little bit later, but Matt, go ahead. What are your thoughts, brother?
I would imagine that since they are in the synonymous regions that are already doing just a little bit of function, when mutations occur there, of course, they're going to have a little effect because they're actually doing very little. So like the first one that you had mentioned, what was that? It's a gene. It's a pseudogene that you mentioned.
Nanog.
Yeah. That one codes for stem cells, right? That one's still functional in mice, but not in humans or chimpanzees, right? So what it's doing would be a functional thing regarding stem cells. We don't have a problems with stem cells in our own bodies. We make them mostly through the femur and the rib cage and larger bones like that. So I would imagine that the reason why it's different in humans and chimpanzees is because the functional role through the process of the embryonic stage is where this gene actually functions. And so when you see like a particular area that's considered a mutation difference between a chimpanzee and a human, with the kidneys, we see that the kidneys function completely different between the two. So what you guys would say is a mutational difference, we would say is a created difference. So it's just the difference between our models. We're saying that this is just an allele difference, not a mutational difference. So
okay, so two clarifying questions there. So first, when you're talking about the functional gene in mice, are you, because not all the pseudogenes or the versions of that gene are involved with the stem cell function. That's what the functioning version of that gene does. But I don't think that might be one of the pseudogenes, maybe, but I don't think it's one shared between chimps and humans. I might be wrong on that, but most of them don't carry out that function. So I guess first, could you clarify on what you meant there exactly?
Okay, no problem. That gene in particular, when it's considered functional, they had a lot of mutations as well in the mice one. It was considered broken, but they said mutations activated it and turned back on, and now it works like it used to originally. We would say no, those mutations were actually functional. That's why they were not really mutations to begin with. We follow mutations in a different view, in a different light. Remember, in our model, it's the nuclear DNA or created differences, and mutations that we see in the mitochondria. These are the regions that would would be caused by the damage from the mutations and what separate us. That's what we trace back.
So you mentioned like kidneys. Are you suggesting that like different organs have different mutations and that affects their different functions? Or I'm not sure I quite understand what you're saying.
No problem. I'll clarify it real simple. Basically, what I'm saying is that when we look into a region and we would say that you guys are like, this is a mutational difference that proves evolution. We're saying, no, it's a created difference and it's functional, because when we're forming our kidneys and when primates are, they both form different types of kidneys. So they're functional regions, as where the evolutionary model would say that they're damaging, right? This mutation is either reached fixation that's beneficial and that's why it's doing it. We would say these are created differences and that's why they're different. That's all.
Well, I think the reason we're pretty sure they are all mutations, though, is because they kind of had the pattern that we would expect if all these mutations occurred. So, for example, there's because different mutations, and I'm sure you're already familiar with this, but because different mutations happen at different frequencies, let's just say they have a probabilistic nature. Like, for example, I can't remember which is which offhand. Transversions versus transitions, for example, like the pattern of going from a pyrimidine to a pyrimidine versus purine to pyrimidine, for example. Like, those obviously happen at different frequencies in the genome. So the frequency that we see that happening naturally is kind of reflected in these different regions, for example. And I don't know if I'm making that clear, but like that's kind of how we're pretty sure that those are mutations, not just created there.
Okay, you're talking about statistical probabilities, and statistical probabilities are great because that's what science is. The more improbable it is, or the more probable it is, the more it's likely to happen. So you're saying that because of the statistical probability of all those things lining up that just so happen to match the evolutionary pattern, it must be true. But then you can counter that argument yourself by saying, well, if we compare this very region over here, like cytochrome C to cytochrome B, we get a different tree of life. So then you have to go back and say, well, maybe it's because it mutated differently, right? So we're saying that, no, these are the created differences because of the beneficial functions that they apply to, because those were never considered in the evolutionary model. That's why they threw them away as junk DNA. And we said, no, they'll have a function. And therefore, that was the real big difference between our models, right? That's what put intelligent design kind of on the map, because, they don't really have too many predictions in their model. We, on Young Earth Creation, we try to have a ton of them. And that was the one that they actually got right, it seemed like. There was no real junk DNA. They're doing transcription. And though it's a little bit, it's still somewhat functional. And they had to say that, well, lots of time has passed, and that's why they gained their function back again. So we're saying, no, that was always their function. There is just a little bit. It's redundancy. It's backup. It's like having a spare tire in your car.
Right. Yeah. Yeah. You're talking about kind of redundancy. Yeah. See, I understand what you're saying, but that's not exactly what I was talking about. If I could share my screen, Donnie, just to help illustrate what I was talking about a little better. It's from BioLogos, and I know you have a bit of a bone to pick with them, but just to kind of illustrate my point more easily. All right. Let me know if you can see it. Okay.
I do see it, but I think once you started to share screen, you started getting a little laggy, but you're good now, Andrew, if you wanted to.
Oh, is my audio still okay?
No, you're good now. So it was just a temporary lag, but go ahead.
Oh, okay. Okay. Yeah. So this is kind of the thing I was talking about. Is there are different kinds of substitutions. So like from pyrimidines to purines, G to C, or A to T, etc. And obviously pyrimidines and purines, so like G and T and A and C, for example, have different chemical properties because they're different sizes, and the chemical properties go with them. So it's more likely that one nucleotide with one chemical fingerprint, let's just say, is more likely to mutate to the same fingerprint, chemically speaking, than to a different one. So for this is why these transitions, for example, where it's going from same to same, pyrimidine to pyrimidine, purine to purine, instead of one to a different one is much more likely, and it's a higher frequency in the genomes of different animals, for example. And you can look at it kind of goes into a lot more detail, obviously, but let's see. Oh, here we go. Between different kinds of organisms, for example, and you see the same pattern, and that's what I was talking about with between transitions versus transversions, and how one's more common in the genome than the other. That's kind of one reason we're pretty confident that these are mutations, because they have that general pattern.
Well, wouldn't that be saying that you just kind of confirmed what we were saying, that if we're finding these mutations in the same region, you're saying, yeah, that should be expected. But that's what we said. You know, we're saying, like, well, if we're finding these and they create a pattern evolutionarily the way that it is, and then you say, well, yeah, it's because the mutations are more, there's more percentages of this happening for that nucleotide going to this one. And it's like, well, I mean, that would explain the pattern then, wouldn't it? That would explain why it would seem
some of it, but that like the particular, I guess there are like more likelihood of one mutation than another, for example, but that's not a deterministic thing. That's not what you would need exactly for like the mutational hotspot idea to work for these different regions, for example. Because the genome is so large that, yeah, of course you're going to have the same ones or similar mutations at some points, but that doesn't really help you here.
Okay, I'll go. I'll answer one more thing, and then we also have a couple other people. But I would say that when we look at the human population and when we see haplogroups arising, we notice that they're pretty infrequent, right? We don't have a lot of haplogroups. We just have the kind of a few letters of the entire alphabet for all different human beings over, let's say, 200,000 years, right? New haplogroups formed. And then we kind of don't see that much anymore because humans have expanded, right? So when we go over and we say, well, let's look at the mutations in chimpanzees, we find a lot more. We find a lot more, I don't think they named them haplogroups, I'm not sure, but we find a lot more mutations and a lot more substitutions in them over that amount of time. We'll just give it 200,000 years. But so what are we seeing though? We're seeing these mutations in these same regions, even though we're split and we're farther away from each other. We're still seeing these mutations come up, rise. They reach fixation in a population, and then there's that new mutation, and we can track, like, oh, that's why they're Native American. They all have Q as a haplogroup. We can trace them. We can see that mutation reach the fixation. So if this was so improbable that it should be happening in chimpanzees and humans, but yet it is happening in them as well. And so it seems to me that there is, even though we're separated, that we're still accumulating mutations in these same regions, and they're still reaching fixations in the same regions that show, like, okay, well, there must be some type of mechanism where these are how mutations occur in those species. So
yeah, I understand the point you're making. I feel like it's just kind of a matter of the size of the genome you're looking at and the likelihood of different mutations, how often they occur. So I'm sure if someone were to look at that and kind of all those factors together, that they would find a clearer answer for that. But yeah, I'll have to look into. That's an interesting point there. Yeah.
And Donny likes to do it, but with ERBs. So that's all him. On
right.
That
Yeah.
I'm not.
Yeah. I've heard him talk about that. Yeah. So yeah.
Okay.
I'll have to do more research on that point.
So I would say, in good discussion, gentlemen, we're definitely getting into the weeds when it comes to synonymous mutations versus non-synonymous, and how common one type of mutation is versus another. But as you pointed out, or we pointed out earlier, Andrew, the fact that the synonymous mutations don't actually change the amino acid sequence, and so the codons can basically change, and but what it reads to doesn't actually change. And so it doesn't really change the actual protein itself, but your non-synonymous changes, they would change the amino acids. and therefore have more of a consequence, would you say, to the organism rather than the synonymous mutations, because they're actually changing the amino acids?
Yeah, they're generally speaking, yeah, like obviously some synonymous mutations are going to have some impact, be it beneficial or negative. Like I know a few papers have, like I think with yeast, maybe. I don't know if that's been debunked by other papers lately. I'd have to look into that, but yeah, I know there have been, yeah.
Okay, so when it comes to these,
off a tangent there.
No, that's fine. That's good. So basically, when you're pointing to these ratios, are you saying because of what occurs on a genetic level with these kinds of mutations, purifying selection should therefore favor synonymous changes more than non-synonymous, because the synonymous mutations don't actually change the amino acids, but the non-synonymous ones do, and there should be a level of favor in the ratios of these mutations, right?
Yeah, I kind of word it differently, but yeah, I generally agree with that. Yeah, there is a ratio you can use there.
Okay. So I think it definitely comes down to functionality, even in your degenerate codons, even in your, Dr. Herman Mays likes to look to statistical hierarchies when it comes to neutral variation, which I think is very similar to what we're talking about here. Right. And I think it's going to come down to the two different starting points of the origin of all genetic variation, right? The evolutionists would say that the majority, or if not all, genetic variation is the result of mutations over time. The creationists would say that the majority, especially with autosomal DNA, is the result of created diversity, designed variation. But we do both agree that mutations happen. And right. I've got 3 references here to non-random mutations and hotspots. Speak to this a little bit. I find the evolutionists might be underestimating the amount of mutational hotspots that actually exist, just how non-random these mutational events are. I feel like they're underappreciating them. Speak to that a bit, Andrew.
So the idea of a mutation hotspot due to some chemical property or another, or just the epigenetic factors involved with it, is that the mutations in one region, or saying it differently, that there are probably going to be more mutations accumulating in one region, the hotspot in this case, than in a different region, a cold spot. I think what we might disagree with, though, is the number of mutations there might be increasing. It may be a higher rate of mutations happening in that particular region, but the mutations are still probabilistic. Like they're not gonna. There are. That's why I don't like the word random as much, and others have spoken to this, because as we've been discussing, there are some statistical likelihoods or biases with one mutation versus another, but they're not deterministic, which is what you would need for a specific set of mutations to occur in a specific region, because again, the genome is so vast and so large, even with the timescales that we're talking about in terms of evolution, that the likelihood that even those biases would account for the same order here is kind of remote.
Firstly, when it comes to mutations, I would argue that from a design perspective, especially from the design diversity perspective, and starting with theology, God, he knows the future. He has divine foreknowledge. And so he knows what's going to happen after creation. He knows what's going to happen after the fall. He knows the various environmental changes that are going to take place. He knows what's going to happen after the flood and the flood bottleneck and the shifts in environments. And so I would argue that a lot of the mutations that are accumulating, they are in more ways deterministic than the evolutionists would say. And so I think God designed ATCG. I think he's designed certain spots in the genome, certain letters to change or mutate more frequently. I mean, just one mutation in coat color in a bear can result in what we see with a polar bear. And so it looks like it's been a big change, right, in its environment, but it's actually due to a mutation. It's due to a point mutation, something that's deleterious, but there was a benefit in terms of the environment. And so I think a lot of this is the result of divine foreknowledge and some determinism at play in the sense of mutations. Go ahead and speak to that a little, Andrew.
Yeah. So I guess one point, and then I do have one more question if you want to wrap this up soon.
Sure.
Would be, it seems like at that point, which I don't disagree with that possibility, because obviously God can do whatever he wants, or any other kind of deity or supernatural intervention can occur. But I think at that point, we're not in the realm of scientific inquiry anymore. And so we can't really investigate it by scientific means. So I would just say that, I guess my question is, since you accept mutations do occur, but created diversity is also a thing according to your model, how would you kind of distinguish between the two? If you were kind of given one side by side, could you tell the difference?
Yeah, that's a good question. I could answer that a number of ways. The first point I want to say is I do believe that there is a level of predictability with what I'm saying. And that would come down to forward-thinking mechanisms. And so if what I'm saying is true, that a lot of these changes point back to a forward thinker, then we could study the changes. We can study a little bit more deeply as to which mutations are non-random, which ones are more random. We could look to epigenetics, basically, where environment can dictate which genes are turned on, which genes are turned off. So in a nutshell, yeah, I think we could predict that when we study the genotypes of the organisms that are experiencing a lot of these mutations and these changes, we could study to see if these are happening by chance or if there seems to be some directedness going on in the genome. You could respond to that in a second, but your second question, but I would say this for disclaimer, the patterns we're talking about here, the nested hierarchical patterns, even when it comes to the so-called neutral changes like third position codon changes, I do believe that they're there for functional reasons. And I do believe, like, for example, this paper here, which I know you're familiar with, redundancy of the genetic code enables translational pausing. And so it looks like rather than these sequences just being able to tolerate error, because I do believe there's a lot of front-loaded redundancy and also over-design in the genome. And so, like, for example, an engineer, when he builds when a group of engineers build a bridge, they don't build it to the exact, they don't build it in a way that holds the exact amount of cars that should hold it, right? They over design it. So if it's supposed to hold 1,000 cars, they'll design it to hold 10,000 cars, right? That's good. That's good forward thinking. And so I think we see a lot of that in the genome. And so just because a region is not conserved does not mean that it's non-functional, or just because it seems to have a neutral change doesn't mean that it's also not functioning in terms of, and what this paper goes into is the dual functional nature of a lot of the code, where you do have these degenerate codons that tolerate error. So they're redundant. That's good. But they also help. Yeah, there's information flow going on. The redundant elements help to slow and speed processes in the cell. And so there's a lot of multifunctionality going on. And I think Matt would agree that we would predict for a lot of these sequences and a lot of these patterns, the reason why we're seeing them is because there's not only one function, there's multiple functions going on, kind of like we see in protein moonlighting, where you have these nuclear DNA proteins that function not in one way, but in multiple ways. I said a lot there. There's one more question I have to answer that you asked, but to be fair, if there's anything you'd like to respond
to, that's okay. I can't even remember the second question I asked. Anyway.
I think it was basically, I think your question was, how can I differentiate between that which is design diversity and then that which is mutation related?
Yeah, that's right. Yeah. I guess before you answer that,
I said a lot.
The only thing The only thing I would say is probably we would disagree on the how to put this, the significance or commonness, I guess, of this this codon bias is what you're talking about in terms of different lineages, because when you look into it, for example, vertebrates or most vertebrates, since they generally, compared to something like a fruit fly or bacteria, have very small population sizes, selection isn't as efficient. So that way you tend to see more of this selection for certain codon biases in bacteria or Drosophila compared to vertebrates, for example. So it's, I would say it's not a one or all thing. So it has to be compared between different organisms, so that might affect it. And yeah, I guess that'd be the main point I'd make there, is it's not a generalization that's that easy to make.
Oh yeah, that's why I think Dr. John Sanford actually, with the genetic entropy, he doesn't really focus on bacteria and mosquitoes and fruit flies and things like that, because their mutation is a little bit different. So he looks at more like the humans. So when you wanted to create and look at the genome, he mapped what he would consider Eve's sequence based on a genome without mutations. And as you know, Dr. John Sanford, his entire life has been basically gene sequencing. He invented the first stages of genetic manipulation with plants in the first GMO plant gene gun. So I would imagine he did a pretty good job at being able to identify what was a mutation and not. He has a whole paper on it, actually.
Well, Matt, how would you, so in a nutshell, when it comes, and it's a great question that Andrew asks, how can we tell the difference between that which is the result of created heterozygosity? Heterozygosity means a state of DNA diversity, essentially, and that which is mutation related. You know, a quick answer is we would look at allele frequencies. We could look to alleles that are very common, where basically everybody on the planet has them. They're some kind of benefit to humans or whatever organism we're looking at. They're not disease causing. We could confidently say that they are, because they have a very high frequency, right? And so we could say that they're created. Alleles that are rare, they exist at very low frequencies. Let's say blue eyes, for example, we would say are most likely the result of mutations. So the real short answer is allele frequencies. But a lot goes into that. I wonder if Matt would have roughly the same answer if he wanted to elaborate. And then Andrew, let's throw it back to you to critique our response.
Yeah, and that's also a good way we can actually determine whether or not the junk DNA has any function at all. Is the body supplying ATP to these regions? If it is, then it probably has a function. Another good way would be, if it mutates, what region of the body does it affect? For example, if the CO1 gene, if you don't think it's doing much other than respiratory reactions, but then you get a mutation in there and it makes you go blind, then you know that there's a secondary function that has to do with your eyes. So I think that we're still learning so much about the genome that it's quite amazing. I like it.
Andrew, go ahead. Take your time.
Yeah, I guess the only thing, that's an interesting point there. I guess the only thing I would say is, in terms of alleles and the kind of translational speed you're talking about with regard to synonymous codons, or synonymous mutations specifically, it seems like, based on my understanding at least, that would affect cellular processes, of course, but I don't necessarily know if that would result in different alleles per se, because the alleles come from, I guess it does go into the nuts and bolts of what you want to define as an allele or not, whether you want to call regulatory portions of the DNA alleles or not. But yeah, I guess that be what I have to say there. I hope that's my point there is a little clear.
I think the bigger picture, and you're more than familiar with the ENCODE project, and if you'd like to say this is something specific to that too, Matt, feel free to do so. But I want, especially for the audience sake, is the bigger picture is it comes down to function. And I would say, because the evolutionary model basically believes that everything is a process of descent with modification, and just millions of years of evolutionary history with precursor ancestors, so on and so forth, which means we should find in the genome a lot of genetic mistakes, as you've pointed to, right? Pseudogenes. So there's a debate there. We should find the ancient remnants of viral infections, ancient invasions of parasitic DNA, basically. That's where we get into ALU discussion, ALU sequences, or ERVs, so on and so forth. We'd also expect just to find a lot of evolutionary leftovers and a lot of non-functionality going on in the genome. We should not only find, if evolution is true, we should not only find nested hierarchical patterns in the biological world within the functional sequences, but we should also find nested hierarchical patterns within the biological world in the non-functional regions. And so the question to the common design proponent is, why do we find nested hierarchical patterns and distributions in sections of the genome that aren't functional? And that's what you'd expect if evolution was true. If it's descent by design, or if it's nested hierarchies by design rather than descent, then we would expect a lot of functionality. So we would find these nested hierarchical patterns within so-called non-functional regions and non-functional elements. We'd actually find those to reflect just that, which is function. And so the one thing I want to say is, in terms of the bigger picture, we understand very little of the DNA language. We're learning so much. The ENCODE project discovered a lot of activity. Yes, the activity doesn't necessarily equate 100% to function, but that's why I think it's so important to study the various interactions between the systems of the cell. Because yes, we find a lot of activity, but we need to look at specific interactions. We need to look at transcription factor binding. We need to look to protein binding to DNA, histone binding, histone modification. Three-dimensional interactions within the cell. And so a lot of this has been looked at, and specifically in the ENCODE project and subsequent projects as well. And we actually do find a lot of interactions taking place, and a lot of these interactions actually do have outcomes and consequences to the function of the cell. And so I think that would tell us that the majority of the genome is functional. But again, a lot of this is dependent upon whether the activity and the interactions going on is significant function. And so, Andrew, respond to that. But my whole point is, the bigger debate question is going to come down to, and I believe it does come down to, function. And that includes the pseudogenes you're talking about, even the ones that don't produce RNA or have a missing promoter. That includes the ERVs that are transcriptionally silent that have been discussed. And the Grayson has pointed to that. And also the so-called third position codon degeneration. So anyways, I said a lot there. Speak to that a little bit. Tell me if you agree with my bigger picture assessment, and then we'll kind of go from there.
Wait, hold on, Andrew. Don't lose your thought. Hold that thought. Donnie, I just wanted to acknowledge there's a few other people here waiting to have a word or two. I'm not sure if I just don't want those guys to get forgotten about. So
sure.
I've been patiently waiting on. All right,
Andrew, go ahead. And then if anybody had any thoughts or points they wanted to make too on the topic of evidence for evolution, feel free to just jump in. Consider it a free for all now.
Yeah, I guess the last thing I want to say, and I don't want to take up too much of the limelight, so I appreciate the time you've given